Welcome to our OTS webinar Guidelines for Experiments Using Antisense Oligonucleotides and Double-Stranded RNAs with David Corey, who's the OTS President-elect, who will be the OTS President in 2020. Just quickly, if you have questions, we will have a question and answer session after the end of the presentation. Please put your questions into the chat box, as unfortunately, if we allow people to speak, the background noise is too much for the presentation. And over to you, David.
Petra, thank you very much. And I'd like to thank everyone who will be listening to this. Today I'm going to be talking about guidelines for experiments using antisense oligonucleotides and double-stranded RNAs. I am first going to talk about the origin of the problem that we face with the design of experiments that are going to be persuasive. And then, in the second part of the talk, turn to a description of guidelines for robust experiments using antisense oligonucleotides and double-stranded RNAs. So this talk is going to focus on two different approaches to silencing gene expression. The first is antisense oligonucleotides. The second is double-stranded RNAs. Both of these are antisense technologies. They both involve a synthetic nucleic acid strand that binds to the sense strand of messenger RNA. Antisense oligonucleotides can act by sterically blocking the progress of ribosomes or by sterically blocking the splicing apparatus. They can also recruit RNase to cleave messenger RNA. Double-stranded RNA, by contrast, uses the endogenous RNAi machinery. The RNAi machinery binds to the RNA and helps it to efficiently locate its target sequence. So these are two different mechanisms, but they share important similarities of recognizing complementary sequences, and both of them involve synthetic nucleic acids. Okay, so the history of this was that back in the 1970s, the concept of using synthetic nucleic acids first became widely known with the publication of the seminal paper by Lessinger suggesting that antisense oligos could control gene expression. Okay, then in the 1980s, the field picked up pace because it became possible to synthesize antisense oligonucleotides routinely. Keep in mind that before 1985 or 1986, it was a historic achievement to synthesize a 20-mer piece of DNA. Starting around 1986, good DNA synthesizers started to become available. RNA synthesis started to become possible toward the end of the 1980s. Being able to purchase oligonucleotides started to become routine. And by the end of the 1980s, you had the founding of the first companies that were devoted to developing nucleic acid therapeutics. Okay, so from there, the 1990s, there was some good news and some bad news. The good news was that there was one approved drug. The bad news was that there were many clinical failures. And one of the reasons for this was that an entirely new field of science needed to be developed around antisense oligonucleotides. And there were many challenges that the pioneers of the field had to overcome. And we're going to discuss that more later. Then, after 2000, RNA interference entered the picture as a therapeutic modality for developing nucleic acids. In the 2000s, there was also steady progress with medicinal chemistry and pharmacology, but there was also a steady stream of new clinical failures as workers in the field continued to overcome the daunting challenges that were posed, again, because they had to develop a new science of drug development around nucleic acids. So now, we're in 2018. Where do we find the state of the art? First of all, nucleic acids can be made efficiently on multi-kilogram scale. That's certainly a landmark of chemistry. Multiple clinical trials are now showing long durations of effects. Monthly or even 3 to 4 times per year dosing is proving effective, and I think that's something that no one would have predicted when the field started. We know with certainty that double-stranded RNAs and antisense oligonucleotides are effective in the liver, and we know that antisense oligonucleotides are effective in the central nervous system. And I think especially in the central nervous system, no one could have predicted that they would have worked as well as they appear to be working. Thousands, probably over 10,000 or more patients have been treated so far. There's an excellent understanding of how to design clinical trials, pick a target, and what can go wrong. And that's all important because that understanding is needed to make sure that the money invested in clinical trials is going to be as likely as possible to have a strong payoff. There's one approved double-stranded RNA drug, and there's several approved antisense oligonucleotides. And I think in 2019, we can expect at least 2 or 3 more. But one thing's certain is that we're going to continue to see unexpected setbacks. And part of why I'm here today is that I think the field needs a reminder about how we minimize these setbacks and maximize the return on research investment, both at the academic level and at the industrial level. And I think this final question is key, is how do we maximize good science that drives oligonucleotide medicine and research forward? And that's a serious question for the field because we don't have the luxury of just kind of meandering forward with a business-as-usual approach. The field needs to move forward as efficiently as possible because it competes with antibodies. It competes with small molecules. And now it's going to start competing with gene therapy. So efficient forward progress is critical if the field is going to continue the upward trajectory, the exciting trajectory of success that we've seen in 2018. Okay, so it's worthwhile comparing oligonucleotides to standard small molecule drugs to remind ourselves what the challenges were when people first began developing these as nucleic acid therapeutics. Small molecules in general are large. Many times they are orally bioavailable. They're under around 500 in molecular weight. They're usually uncharged. Antibodies are larger. They've also been very successful in the last decade or so. They function mainly extracellularly. Now, oligonucleotides, by contrast, have to function within a cell unless they're an aptamer, and we're not discussing aptamers today. But to enter the cell, they have to enter it in spite of being highly negatively charged. So a large molecule, negatively charged, I think that underscores why a new understanding of how therapeutics might be developed was needed for oligonucleotides. So I've just finished explaining to you why small molecules and nucleic acids are different, but let's also not forget that a synthetic drug is a synthetic drug, and many of the considerations are also going to be the same. So the oligonucleotide field has historically benefited from workers in the field who were familiar with how small molecule drugs were developed. So both small molecules and oligonucleotides must bind to a target. Okay, that means there's going to be on-target interactions that produce the desired results. For example, controlled gene expression to affect the disease favorably. But there's also going to be off-target interactions that are going to produce unwanted side effects. And you're always going to get off-target interactions with any synthetic molecule. The key is that you want the difference between on-target effects and off-target effects to be as wide as possible. Okay, that's the therapeutic window, and that marks a successful drug development program if the size of that window is something that works for patients. Okay, so small molecules and oligonucleotides, they make different interactions. They're different kinds of molecules, and they cause different off-target effects. But these off-target and on-target effects have to be balanced. Therefore, standard pharmaceutical development, common sense, also applies to oligonucleotides. Okay, so that's another way of saying that the same high level of professionalism that big pharma employs when they're developing drugs needs to be employed during the development of oligonucleotides. And I think that the companies that have been the most successful at developing nucleic acid therapeutics, have certainly shown us all that level of professionalism is possible in our field. Okay, so to kind of review that in graphic form, you start with a synthetic molecule. The fact is that if you put a synthetic molecule into cells or in an organism, you are always going to get a phenotype. That is a certainty. Now, what you hope is that phenotype is due to an intended target. But there are many other unwanted interactions that are possible inside cells that are also going to produce a phenotype. So what are the possible sources of those interactions that may produce off-target phenotypes? Well, if your oligosynthesis isn't pure enough, you might have impurities that cause toxicity. Now, taking it to the next level, even if your molecules are pure enough, they may bind to cell surface receptors, and those may start a signaling pathway that causes off-target effects. You might also have binding between the negatively charged oligonucleotides and proteins inside cells. So it's not hard to imagine that a negatively charged oligo will naturally seek to bind to positively charged proteins, and that those interactions might trigger normal cellular responses to DNA or RNA, or interactions with proteins that don't normally bind nucleic acids. They can be hard to predict. Now, the basis for using DNA or RNA is, of course, the potential for Watson-Crick base pairing for a target. Another source of off-target interactions is binding to non-target DNA or RNA. Now, certainly that can be minimized by appropriately searching for non-target sequences and minimizing that, but it's always going to be possible. You can have the induction of immune-responsive or interferon-responsive genes. And finally, there's always the potential when using double-stranded RNA that one might crowd out normal RNAi pathways. Now, I'm not sure that anyone knows how big a danger that is, but it's something to always keep in mind. Okay, now what I would like to emphasize moving forward is that none of the considerations that I've talked to you about in the last few slides, nor many of the suggestions that I'm going to make in the rest of the talk, are necessarily new. When I was beginning in this field in the 1990s, there was a journal called Antisense Research and Drug Development. It was the forerunner of today's nucleic acid therapeutics. And I had the benefit of being able to read reviews and commentaries from, basically some of the great pioneers of our field. Up here you have Cy Stein, Art Krieg, and Stan Crook. And these are all commentaries that I've posted here that are as valuable to read today in 2018 as they were when they first came out 20 or more years ago. My purpose today is to reiterate some of the lessons that these pioneers taught us, and also to update them somewhat so that this conversation of how to do the best experiments can now be moved into the second decade of the 21st century. Okay. So these great reviews, these great commentaries appeared 20 years ago. Why do we keep having a problem with how people design their experiments using antisense technologies? I have a few thoughts on that. One is that the recognition of a modified nucleic acid and how it works, how it is able to recognize RNA is predictable. It's simple Watson-Crick base pairing. Therefore, it's very easy for someone to fall into the trap that because recognition predicts binding will occur, that the observed phenotype is because the binding occurs. Another reason is that because nucleic acids are such powerful tools, so simple to use, that new investigators who are trained in other areas of science are drawn to use them. And I think, understandably, those new investigators will not have read the old literature in our specialized field that dates back to the 1990s. Another reason is that, let's face it, there are very few incentives for obtaining negative results, but there's very powerful incentives for obtaining positive results. That's another way of saying that it's easy to not do enough controls, see the result that you want, move forward, and publish your results. And because a lot of reviewers out there are not familiar with ways in which to design appropriate experiments with nucleic acids, those results end up getting published in the peer-reviewed literature. And I hope that one of the outcomes of this conversation that we're starting today will be a new 2018-2019 version of the minimum guidelines for nucleic acids that can be used to advise authors, reviewers, and editors as they decide what should be in the peer-reviewed literature. There's also been an enormous increase in potentially important nucleic acid targets, so many more therapeutically important messenger RNAs. We now have the world of microRNAs, as well as the growing world of long non-coding RNAs out there. So, is this really a problem? I'd like to give you one case study for a single long non-coding RNA. I want to say at the outset that I don't mean to be picking on the people who have studied this non-coding RNA. There are many microRNAs or long non-coding RNAs that have similar problems in what's been studied with them. This happens to be the first one that I actually looked at and researched. It's a long non-coding RNA called UCA1. About a month ago, I searched on PubMed for this RNA, and about 228 relevant papers have been studied over the last 4 or 5 years. So I chose to analyze relatively closely about 49 papers from 2018 that studied this long non-coding RNA. Almost all of these papers used siRNAs, shRNAs, or microRNA mimics to study the action of this non-coding RNA. Now, none of these articles had adequate controls, so none of them lived up to the minimum standards that were outlined in the 1990s or that I'll outline in a few minutes. And what is the consequence of that? Well, these 49 papers ended up suggesting that this non-coding RNA was responsible for this extremely wide array of different diseases. There are many different types of cancer, but there are also diseases like cardiomyocyte hypertrophy and epilepsy that aren't involved in cancer. So if you were to look at this literature, you would believe that this non-coding RNA is involved in dozens of different diseases. And what's more, it appears to be, from the literature, many different microRNAs are involved in the control of this long non-coding RNA. It's been reported to be able to induce microRNA expression, be a target for microRNA expression, or act as a microRNA sponge. So I make no claims that I'm drawing any conclusions about the individual papers that studied UCA1. It's entirely possible that some of them are quite accurate. However, taking this literature as a whole, it appears that UCA1 is involved in an implausibly broad range of processes, and that it's affected by an implausibly broad range of microRNAs. And this is compounded by the fact that the controls in these papers are inadequate. In addition, as a rule, the characterization of the basic molecular mechanism of long non-coding RNAs is also lacking in these papers. So where does that leave us? Well, we have a long non-coding RNA. It might be important for disease and therapeutic development, but because we have so many studies where the controls are inadequate, if there were good studies, they're obscured by the mass of studies that are perhaps not so good. So one of the purposes of starting this conversation about guidelines would be to help readers, help editors, narrow down the papers that are worth attention, and also to help authors produce papers that are going to be more persuasive. Okay. So now to turn to the second part of the talk, guidelines for robust experiments using antisense oligonucleotides and double-stranded RNAs. So we'll start with guidelines for cell culture experiments. We'll briefly talk about guidelines for animal experiments, antiproliferative phenotypes because there's an extra need for caution there, and then we'll conclude with general considerations for experimental design. Okay, so some general comments first. One of the great advantages of our field versus other fields that we have not taken full advantage of is how easy it is to obtain controls. It's usually going to be simple and relatively inexpensive to test negative control experiments, that is, compounds that are not complementary to the target. Okay, so all you do is go to your oligonucleotide supplier, design compounds that aren't complementary, and just fit them into your testing protocol alongside the oligo that you believe is going to have some kind of on-target activity. In general, it's also very easy to obtain a positive control oligonucleotide that controls expression of some kind of benchmark gene. So for example, in my lab, when we're starting off with a new target, we often like to look to make sure that we can inhibit the non-coding RNA MALAT1, just to make sure our transfection protocols are proceeding appropriately. We use a known oligonucleotide, make sure we're getting efficient knockdown of a gene that's well characterized. And finally, it is almost always going to be possible to obtain more than one active compound against your target. So it's obviously an advantage if you're looking at a phenotype to make sure that you have 2 or 3 compounds that knock down your target, and then several compounds that are not complementary that don't when you're evaluating what phenotype is. Okay. Now, throughout this discussion, we do have to recognize that experiments require resources. And I think we all recognize that there's a balance between the best possible experiment that you can do and the resources that you have available. There's going to be one set of resources available at a company like Ionis or Alnylam, who are able to throw hundreds of oligonucleotides at a particular problem. There's an entirely different set of resources at a small laboratory that may be working off a budget of a few $1,000s a year. So the goal here is to do the best experiment with the resources available. So what I'm going to be trying to do here is sort of outline what the best experiment might be, but also at the same time what the absolute minimum experiment should be in order to have a paper published in a peer-reviewed journal. But because I'm suggesting minimum experiments, that doesn't mean that they're the ones that an experimenter should necessarily do if they have more resources. Because in most cases, experimenters should try to do the most rigorous job possible and make the strongest possible case for their conclusions in order to satisfy or convince reviewers. So keep in mind as I move forward, we're talking about a minimum standard where hopefully if it doesn't meet the minimum standard, papers would be editorially rejected without review. And then, to the extent that resources allow, being more persuasive on top of that to fully convince the audience of the strength of the results. Okay, so guidelines for cell culture experiments. What's your first step? Your first step is that you need to discover an active compound. If you don't have a compound that's active, there's actually not much point in doing a lot of controls. You want to see something that's exciting first. Typically, it would be best if you identify several active compounds. You can then choose the most potent one to investigate further. So in my academic laboratory, typically we might test 6 to 10 compounds to begin with if we're looking for an agent to change gene expression, and then we'll pick the 2 or 3 best to move forward with after that. Okay, step 2. Once you've chosen your active compound, you have its sequence. You can then design controls based on that sequence. Now, typically, one type of negative control that's useful is to design 3 to 5 mismatches into the sequence of the active oligo. Okay, that should abolish binding, but it keeps most of the molecule intact. If a double-stranded RNA is used, some of those sequences, mismatches should be within the seed sequence. Step 3 is related to step 2. Design a scrambled control where you simply move around blocks of nucleic acids around the original parent sequence. And then typically test both the mismatch and the scrambled control alongside your parent active oligonucleotide. And then finally, step 4, choose at least one other active oligonucleotide from the ones identified in the initial screen. So what you're looking for at the end of the day is that this compound should have the same ability to knock down the target and produce the same phenotypic effects as the chosen lead compound. Okay, let me just give you an example from my own laboratory of how we approached using these controls for a challenging problem. This involves a disease called Friedreich's ataxia. It's one of the most common inherited ataxias. It's a devastating neurodegenerative disease. It's currently incurable. It's caused by a deficiency in expression of the frataxin protein. It's caused by an expanded GAA repeat within intronic frataxin messenger RNA. So this is a disease that's caused by a mutation within a non-coding RNA. The frataxin protein itself is unchanged. The amino acid sequence is the same. But there is lower expression of frataxin messenger RNA and protein. Therefore, this is a lack of function mutation, loss of function, and increased frataxin protein would almost certainly correct the defect. So how do nucleic acids, how can they be applied to Friedreich's ataxia? Well, first, this is an unusual challenge. The target's within an intron, and we would have to manage to upregulate protein expression. So whenever you see a target with this kind of novelty, the bar is going to be higher to showing, to building a case for your audience that any phenotype that you see is an on-target. There is likely to be an on-target effect. Okay, so why are frataxin levels reduced in Friedreich's ataxia? The thinking in the Friedreich's ataxia field is that the expanded repeat binds to chromosomal DNA and forms an R loop. That R loop acts as a brake on transcription and reduces gene expression. So our hypothesis was that we could target the expanded repeat with either double-stranded RNA or antisense oligonucleotides, block the mutant repeat, and prevent the RNA from binding back into the chromosomal DNA. No R-loop formation, no applying the brake on expression. Frataxin expression should go back to normal. So here's our example of how this kind of applied. We designed double-stranded RNAs to target the repeat within frataxin. It was challenging because activation was mechanistically novel. The target was an intron, and activation was only threefold. This last one is something I think for you all to consider. If you're seeing a 5, 10, 20, 50-fold difference in gene expression when you use a synthetic oligonucleotide, it can be much easier to tell the difference between an off-target effect and a non-target effect. In this case, we were dealing with something that on a good day might be 4-fold, but on a bad day might be 1.5 or 2-fold. And the practical consequence of that was that many of these experiments needed to be done many times before we had the confidence that a 3-fold level of activation by a novel mechanism was true. In any case, we analyzed both protein expression by Western analysis and RNA expression by quantitative PCR. And I'll point out another experimental tip is that when trying to convince people about a novel mechanism, showing both RNA and protein levels, can be very helpful, especially when the protein level is what's ultimately going to be needed for therapy. As you can see, we used both a scrambled oligonucleotide and one with a mismatch control. We also used oligonucleotide with no complementarity. We used a positive control that targeted frataxin messenger RNA. And what you can see with SIGAA and SIAAG is that there were 2 compounds that were able to activate gene expression. So in a nutshell, it shows everything I was just telling you about. Scrambled control, mismatch control, positive control showing our transfection was working, and then 2, not 1, but 2 active compounds that upregulated gene expression. When you're building a case for your mechanism, it's usually good to apply some mechanistic data. What I just showed you was that we were using double-stranded RNA. Double-stranded RNA associates with its target in complex with Argonaute protein. So in this particular case, one mechanistic experiment to do was RNA immunoprecipitation, showing that Argonaute 2 was recruited by the synthetic RNA to the intended target. And what you can see here is that the double-stranded RNA did in fact recruit Argonaute 2 to the target. Remember I told you that the mechanism appeared to be involving R-loop formation? The prediction would be that adding our synthetic RNA would reduce R-loop formation. So R-loop formation is monitored using an anti-DNA RNA antibody, and this experiment showed that we were indeed reversing R-loop formation. So the point of doing those mechanistic experiments is to provide multiple lines of evidence that your compound is functioning by the mechanism that you propose. And if it is doing that, that supports the belief that it's functioning through an on-target effect versus an off-target effect. Now, the experiments we do should be robust. That means that we should be able to reproduce them in different cell lines. In this case, we were asked by the people at the Friedreich's Ataxia Research Alliance who were funding the work to obtain different patient-derived cell lines with different numbers of GAA repeats and find out whether or not our approach would work in those other cell lines. And what you can see here is that our approach is robust enough to function in every different cell line. Okay, also notice here that we did not use the full suite of controls for all of these experiments. Okay? You don't need to do that every time. Once you've established that the on-target effect is occurring, you don't need to devote resources to doing them in every downstream experiment. Again, this is the difference between doing the best experiment, where you would want to use all those controls, but realizing that you come up against constraints of time and money, and that there's just not a lot of value for using those controls once you've already demonstrated a non-target effect. Okay, finally we moved on to do something a little bit even more novel, which was to obtain single-stranded silencing RNAs from Ionis Pharmaceutical to see that these compounds, which are single-stranded but function through the RNAi mechanism, could also act to activate frataxin. And from this study, I just chose you a dose-response assay. Now, first of all, you can see that the dose-response assay works. We get good potencies, and that's important for considering which of these compounds might be candidates for drug development. But another very important point for the discussion that we're having today is that a dose-response profile is more evidence for the robustness of the approach, that you are trying several different concentrations, and you are finding out that some concentrations are too low to work, and that several other concentrations produce a maximal effect. Okay? A smooth dose-response profile also supports that your compound is functioning through an on-target effect. And this just shows with a different type of chemistry, a bridged nucleic acid, that we get the same thing. The point here is that by using two different chemistries, and now we're using antisense oligonucleotides, these are functioning not through an RNAi mechanism, we get upregulation of frataxin in both cases. So now we're using double-stranded RNA, single-stranded silencing RNA, antisense oligonucleotide, three different classes of antisense technology, two different antisense mechanisms, but they're all producing the same result. So all of these pieces should be building your confidence that the activation of frataxin, again, a novel target, is occurring through an on-target mechanism. The final thing to look for when you're considering whether the research that you're evaluating is convincingly demonstrating an on-target mechanism is to ask, are follow-up papers being seen in the literature? Now, it's best if those papers are occurring from multiple laboratories, but you at least would like to see them coming out of the laboratories that originated the invention or the technology. And in this case, for this, we published the original paper in 2016. Two papers have appeared in 2018, and we're hoping that we'll be able to publish one additional paper looking at activation in neuronal cells in 2019. So again, when you see papers following up on the initial discovery, you can feel a little bit more confident that the result is real. So finally, to close out this kind of section, I'd like to say a little bit about the responsibility of oligonucleotide suppliers. We all recognize that suppliers do heroic things for us, that contemporary biological research would not be possible without the suppliers of synthetic nucleic acids, who, for most of us, at a relatively inexpensive price, supply us with what we need quickly and with very high quality material. However, for many knockdown experiments, antisense oligonucleotides, and to a lesser extent, chemically modified duplex RNAs, can be so costly, maybe at the price of several $100s each one, that well-controlled experiments are actually discouraged. It becomes difficult to actually afford the control experiments that I was telling you about. So one of the proposals that I would have that the field discuss is that suppliers should make sure that they ensure that oligonucleotide packages are being sold with suitable controls that are available at affordable prices. Because price should never be an excuse for not using minimal controls in cell culture. And what I would foresee would that this would actually be a win-win solution, that investigators would get a better package of oligonucleotides, and that the suppliers would end up selling more oligonucleotides for more money anyway and making more profit. So there should be a way to work this out. But price should not be a limitation on using the minimum number of controls necessary for an experiment. Okay. Sometimes I have discussions with people about the value of mock or transfection agent only controls. I would say that these controls are not a useful use of an experimenter's time, especially one who's experienced at using transfection reagents. My laboratory's experience is that when you put a transfection agent into the mix with an oligonucleotide, it almost always produces many more off-target effects or toxicities than the transfection agent or the oligonucleotide alone. So if you want an apples-to-apples comparison and you're using a cationic lipid transfection reagent, for example, the best thing is to have that in combination with a non-complementary nucleic acid that's similar to the one that you're using as your agent. Okay, so that was cell culture. What about animal experiments? Animal experiments, we start to run into this problem of resources more seriously because you're going to need much more nucleic acid, and the animal expenses might be high. So in theory, the requirement for controls is the same in animals as it is in cell culture, but we have to recognize that for many people, resources are going to be limited. So recognizing that, someone who wants to do animal experiments needs to have done thorough and convincing cell culture testing first. And then in the animals, at least one control needs to be used for the animal testing. Obviously, an adequate number of animals needs to be used. In too many cases, in published work using antisense oligonucleotides and double-stranded RNAs, the numbers of animals used has been too few to be thoroughly convincing. If possible, knockdown of the target gene expression should be evaluated, and if phenotypic endpoints are available, they should be monitored as well. Okay, so where are we now? So gene knockdown is routinely achieved in the liver. So if you want to go after a target in the liver, I would say the bar is a little bit lower in demonstrating convincingly that you're knocking it down. But success in other organs has been much less precedented. In particular, targeting tumors or non-hepatic tissue outside the central nervous system, I would say requires more evidence. In addition, we know that oligo gets delivered to the liver. If you're going beyond the liver, it would be very, very helpful to do experiments that also demonstrate that you're getting distribution to target tissue. Okay, related to targeting cancer, antiproliferative phenotypes. This is a long-standing problem back to the early 1990s for nucleic acids. Because cancer is such a big area of research, many experimenters have applied antisense oligonucleotides and double-stranded RNAs to cancer research. Reduced cell or tumor proliferation has often been the goal. The problem with that is that virtually any oligonucleotide or double-stranded RNA, if used at high enough concentration, can reduce cell proliferation. In addition, if you were to screen a number of oligonucleotides, it would not be difficult to find several that potently reduce cell proliferation through off-target effects. Moreover, even if you are getting an on-target effect, the reduced proliferation that you see can affect many genes, including the target gene. Therefore, just monitoring proliferation or expression of the target gene can be misleading. As a result, decreased proliferation may not be an on-target effect at all. And you'll see if you go back and look at the 1990s papers that I mentioned earlier, this was widely remarked on back then. So what can you do if you have an antiproliferative phenotype that you wish to pursue? Well, it is absolutely critical to adhere to the minimum guidelines for control oligonucleotides. That you should show that several oligonucleotides that are complementary to the target reduce proliferation, while as closely related oligonucleotides that are not do not reduce proliferation. So multiple oligonucleotides absolutely must be used. For animal experiments, there should be some effort to establish that tissue uptake is occurring. And also, in that tissue, there should be demonstration that a benchmark gene, which does not affect tumor proliferation, is knocked down, showing that you can get an active antisense oligonucleotide or double-stranded RNA into the tissue. Okay, and then finally, if it's possible, if the antiproliferative phenotype is not obscuring gene expression, demonstrate knockdown of the target gene. Okay, finally, let's begin wrapping up. General considerations for experimental design. Experiments need to be replicated sufficiently to ensure reproducibility. But just as importantly, the number of replicates need to be noted and possibly shown in supplementary information. You need to convince your reader that your experiments are robust and reproducible. Gene knockdown should be quantified. Statistical analysis provided. And you need to be very wary of any kind of statistical analysis that claims that a 10 or 20 or 30% level of knockdown or phenotypic change is actually sufficient. Dose response curves that demonstrate potency, very useful and very reassuring to the reader. If a change in protein is desired, that protein expression data should be included. That's something that you should look at. Western analysis, very useful way to show protein expression. The reason for that is that you see a band on a gel, it's primary data, and that band should appear at the correct molecular weight for the protein. It reduces the chances that any measurement of protein is artifactual. Quantitative PCR should adhere to accepted standards. I don't want to go into a long discussion of quantitative PCR. If you are not aware of the ways in which quantitative PCR can mislead an experimenter, you absolutely have to educate yourself. As with our frataxin research, if a result is entirely novel, it should be replicated either by another experimenter within the laboratory or by another laboratory. So with our frataxin results, 5 or 6 people in my lab have been able to replicate that. Certainly reassures me as the PI that the result is real. Okay, microscopy. You often see experiments with fluorescently labeled oligonucleotides. There is no doubt that those experiments can be useful. But let's also keep in mind that they can also be misleading because the fluorophore is a large, generally quite hydrophobic molecule that can change uptake or tissue distribution. Also keep in mind that at the end of the day, you want to see a change in gene expression. So you have to keep in mind in perspective, how important those experiments with fluorescent oligos are going to be. Finally, for double-stranded RNAs, 5' race can be a very useful piece of evidence about mechanism, especially in tissue, demonstrating that your intended synthetic RNA is inducing cleavage of its target RNA in the target tissue. Now, remember, that cleavage can be at a very low level. It doesn't necessarily mean that efficiency is high enough to produce the phenotype, but at least it shows that your agent is getting into your target tissue. It gives you somewhere to start. Okay, so you've done all your controls. What then? Well, one other thing that I would propose as a minimum that people in this field should do is actually explain to the reader what your controls are. So several years ago, my laboratory published a complicated piece of work, and you can see the schematic up here, showing that a double-stranded RNA targeted to the promoter region of the cyclooxygenase 2 gene could bind to a non-coding transcript and not only activate expression of cyclooxygenase 2, but also expression of the neighboring gene, PLA2G4, that encodes an enzyme that makes COX-2's substrate. Okay, this was a very novel hypothesis that was certainly very, very novel to the reviewers and encountered actually an enormous amount of pushback from the reviewers in ways that were not always extremely well reasoned. So finally, this paper eventually was published in Nucleic Acid Research. And the editors there wisely advised me that it was time to publish the paper, but that what I should do was actually collect the text spelling out what the controls were and have them there so that they would be available in print to not only directly rebut the reviewers, but to be there to reassure the reader that we'd done the best possible experiments and built the strongest possible case that our work was valid. And as you see on the next text, this is the text taken from our paper where we centralized our explanation for why we believe that our reviews were sufficient to uphold our conclusions. So doing this is very easy. It doesn't take much time to write a couple paragraphs describing your controls. And by having them there, you make the best possible case for why your data is solid. The reviewer doesn't have to guess. The reader doesn't have to guess. And you can summarize in one place why your data has met the minimum standards of controls that I hope that this conversation that we're starting today will end up requiring for papers in this field. Okay, in summary, nucleic acid therapeutics have a long history dating back to the 1990s of unreliable reports. There's been an equally long history of outstanding commentaries suggesting how to correct that. And I think it's time to get back on the path of discussing those commentaries. There's no doubt there's a trade-off between the best possible experiment and what your average laboratory has in terms of resources to accomplish them. We don't want to prevent experiments from being done by demanding that experiments be perfect. However, experiments should follow the minimum guidelines for control oligonucleotides. At the end of the day, though, when we're talking about on-target effects versus off-target effects, the responsibility goes both ways. Readers have to be skeptical, and authors have to be persuasive. And hopefully, if those two things meet, we'll have a successful partnership and we'll have solid papers. So finally, just to summarize some of the suggestions, there should be some general textual requirements. The source of a hypothesis should be clear. Data that argue against cherry-picking a hypothesis and that the hypothesis was, how the hypothesis was selected should be clearly presented. So too often, papers will just start with, we decided to focus on microRNA X, or we decided to focus on long non-coding RNA Y. But the reasons why they started there and why that, with that long non-coding RNA or that microRNA that ended up 7 figures later being so exciting are often unclear. Source of the hypothesis has to be defined. The evidence suggesting that an observed effect is on target should be plainly presented in the text. Experiments aren't perfect, right? As an editor, all I ask is that the experimental limitations and uncertainty be candidly presented. In addition, potential alternative hypotheses that may explain results and that do not involve on-target engagement should be acknowledged. Okay, this gets to a philosophical point. In science, we are not trying to prove a hypothesis. We're trying to supply enough evidence so that the hypothesis is shown to be worth discussing by the community. We're not trying to supply absolute proof. And if there's an alternate hypothesis, it often helps to simply acknowledge it. Okay. So those are easy things that one can do for the text. For cell culture, what I would propose as minimum controls is 2 independent silencing duplex RNAs or antisense oligonucleotides for a target. 1 mismatch control, 1 scrambled control. Those may not be the ideal controls. There may be better ones, but those would be the absolute minimum that one would need for an experiment aiming to modulate gene expression. For animal experiments, the cell culture experiments need to be shown to be robust and thorough. That should be demonstrated. There should be 1 mismatched and 1 scrambled control, or 1 scrambled control used. More controls might be necessary to demonstrate an antiproliferative phenotype. So what I've tried to do here is really provide the easiest possible guidelines for authors to meet. They require only slight rewriting of the text or the use of a few additional control oligonucleotides in experiments. I don't believe that this would entirely solve the problem, but I do believe that it would level the playing field, raise the bar, and help make results in our field more reliable. So finally, I would like to thank all of you for taking time out of your afternoon or evening or wherever it is where you are. I really appreciate it. And I would be happy to entertain any questions. I think all you need to do is type them in. Thank you.
Yes, if anyone has questions, please type them into the chat box.
Well, I would certainly be happy to take any questions offline. You can email them to me. How many technical replicates do you suggest are a minimum? That's a great question. Our frataxin work is a good example of how I'm going to answer that question. In that case, the difference between the unactivated and the activated state was at best only about 3 or 4-fold. Because the difference was relatively slight, many more replicates, both technical and experimental, were necessary. So if you're looking at a small change, you're going to need relatively more than if you're looking at a big change. The bottom line is that you need to convince yourself that the result is real. So you have to do as many technical replicates as necessary. Now, if we're doing quantitative PCR, typically in an experiment, we'll do 3 per experiment. Okay, next question. How widely accepted useful are the C911 controls for siRNA? I'm not familiar with those. Again, I think it comes down to there's no hard and fast rule about what you use. You should be able to write a paragraph outlining why you feel those controls that you're using are enough to justify people taking your work seriously. And again, I should emphasize that when we talk about controls, when we propose guidelines, the purpose is not to micromanage people's experiments. There will be many ways of getting to the level where you are doing convincing research. But you will have to explain it. Okay, what would be the minimum number of mismatches in the control oligonucleotide? We generally want to have at least 4. to make sure that we completely eliminate its ability to bind to its target. Typically, because we have a spectrophotometer that allows us to calculate melting temperatures, we'll also look at melting temperatures to make sure that we've abolished any melting to the target, any binding to the target. Okay, what set of control would you recommend for an antiproliferative phenotype? Again, at a minimum, I would say that one that is mismatched, one that is scrambled. And, you know, you may even want to use more depending on how concerned you are about building a case that your phenotype is due to your on-target. Mismatches in the central block or the wings, thanks. If it's an antisense oligonucleotide, we spread the mismatches out throughout the sequence. If it's a double-stranded RNA, we also spread them out, but we make sure that we have plenty of mismatches, a couple of them in the seed sequence. What is your opinion of on-target but non-functional oligonucleotides as controls? I mean, I wouldn't really view those as controls, but you know, they are kind of part of your experiment. You know, for example, let's suppose you screen, if you're an academic lab, you screen 10 oligos. If you're Ionis Pharmaceutical, you screen 100. Many of those aren't going to work. I would like to see that initial screen as supplemental information. I think it's very valuable information.
Have we got any more questions then, or are we all finished with them? One more.
Where's the question?
Oh, it's just saying somebody's typing something, so.
Okay. So once again, while we're waiting for that, if anyone wants to contact me, or if anyone have any comments about this, we are working towards a commentary that will summarize some of these points. And we'd certainly like any input that we have.
Well, I guess, oh yes, there's one more. One more question for you, David.
Is it okay to use universal negative control antisense oligonucleotide? Yeah, I mean, that's certainly a good control to use, and it can be valuable, but I think, you know, once you've settled on your on-target compound, what would be best is actually to make a couple additional controls that are based on that sequence.
Yes, we will finish off here then, and thank you very much, David. That was really informative. For everyone else,
well, thank you all very much. And thank you, Petra, for organizing this.
Yeah, the next webinar will be on the 12th of February on the pitfalls and problems of microRNA expression analysis. Unfortunately, we have to reschedule due to illness of the speaker, so that will be on the 12th of February now. Thank you very much for all attending. And yes, the presentation will be made available